Welcome to Section 6 of Lab Techniques. In this section, we'll be discussing CRISPR-Cas9 and molecular cloning. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's kind of a mouthful. It's a long name that you don't need to remember, but you should know that CRISPR-Cas9 is a feature of the bacterial immune system that allows bacteria to fight viruses. Biologists have discovered that this tool can also be used very precisely to edit the genome. So CRISPR-Cas9 is a bacteria-derived genome editing tool. Cas9 is the endonuclease component of the system that unzips and cleaves the double-stranded DNA. A guide RNA, also called gRNA, directs the Cas9 enzyme to a complementary DNA target sequence and a cut is made. The cell attempts to repair the gap region through non-homologous end joining, which is prone to errors. Alternatively, a genetically engineered sequence of DNA can be inserted into the gap region to circumvent the error-prone process of non-homologous end joining. Let's pull up a figure so you can see what I'm talking about. This is a figure of CRISPR-Cas9, which can be found in Section 6 of Lab Techniques. Notice that a guide RNA molecule right here is shown in the center of Cas9. This guide RNA directs the Cas9 enzyme to the double-stranded segment of DNA that is complementary to the guide RNA. Notice that AUGC on the guide RNA is complementary to the target sequence of DNA. So A pairs with T, G pairs with C, and so forth. From here, Cas9 makes a cut in the target sequence of DNA. Notice that there is now a gap region shown right here. Finally, the gap region can be repaired through non-homologous end joining, or genetically engineered DNA can be inserted. You can imagine that this technology has many applications, including selectively removing abnormal segments of the human genome. However, the technology is currently in its infancy. Hopefully in the coming years, we'll begin to see the application of this technology in treating genetic disease. Okay, now let's discuss molecular cloning. Molecular cloning is a lab technique that allows us to insert a eukaryotic gene of interest into bacteria. In the first step, eukaryotic mRNA is reverse transcribed into cDNA. The cDNA is inserted into a plasmid containing an antibiotic-resistant gene. The recombinant plasmid is then inserted into bacteria. The sample is cultured on a medium which contains antibiotics. Bacteria that successfully absorb the plasmid will survive, and those that did not properly absorb the plasmid will die. Okay, let's pull up a figure so you can see what I'm talking about. This is a figure of molecular cloning which can be found in Section 6 of Lab Techniques. Notice that the eukaryotic mRNA right here is reverse transcribed into complementary DNA, or cDNA, right here. This can then be combined with a plasmid. Notice that the plasmid contains a gene that encodes for antibiotic resistance. The recombinant plasmid can then be inserted into bacteria. You can see that happening right here. Finally, this is cultured on a medium containing antibiotics. Notice that one of the colonies is X'd out right here, and this one is not. This is because not all of the bacteria will successfully absorb the plasmid. Those that were unsuccessful will not contain the antibiotic-resistant gene and will die. In this way, we can select for only the bacteria that successfully absorb the plasmid. Okay, let's do a question. A pharmaceutical company is attempting to synthesize insulin for use as a commercialized medication. What lab technique would most likely be used to achieve this goal? Okay, pretty straightforward, but this question helps illustrate the application of molecular cloning. So the lab technique that will most likely be used is molecular cloning. Insulin used to be purified from pigs and was the only available insulin for diabetics. However, in the late 70s and early 80s, scientists discovered that synthetically derived human insulin could be synthesized by bacteria. It's pretty incredible to think that today this is how most of insulin is made. Obviously, the technology has multiple other applications, but the ability to synthesize human proteins through molecular cloning has many therapeutic applications.